Hello, my name is Julika Huber, and I'm a plastic surgery resident and a postdoc at Dr. Longika's lab. And today I want to present to you my study about the osteoskeletal regenerative ability of microvesicles that are derived from human adipose tissue-derived stem cells upon inhibition of TGF-β signaling. In surgery, reconstruction of bone defects is very challenging due to its poor self-regenerating capabilities and limited therapeutic options. These therapies or these options include autologous or allogenic transplantation or prosthetic material. Drawbacks of these therapies are limited availability, donor site mobility, and high treatment costs, so other approaches are very much wanted. Mesenchymal stem cells have been successfully applied in bone tissue engineering and has been shown that they increase mineralization and osteogenic gene expression in vivo and in vitro. Initially, it has been thought that they exert their regenerative effects by differentiating at the injury site. However, it has also been shown that after about 48 hours, the cells die. And now it is thought that paracrine signaling via MSC-secreted factors might be the more likely mechanism of regeneration. MSCs produce and excrete microvesicles and exosomes that carry microRNA and proteins that are now thought to mediate the regeneration at the target site. So in our study, we collected human adipose tissue-derived stem cells and treated them with SB431542, which is a TGF-β inhibitor. TGF-β holds a very complex role in bone regeneration. And in a recent study in our lab, we showed that TGF-β inhibition leads to increased expression of osteogenic markers in bone regeneration and bone defects in vivo. So after 6 days of osteogenic induction of our hASCs with and without SB treatment, we harvested these microvesicles via ultracentrifugation. We then characterized the microvesicles via nanoparticle tracking in Western blot, and then we continued on with our osteogenic in vitro assay. So we cultured osteogenically induced human hASCs with microvesicles that are derived from either SB-treated or untreated hASCs. And we then also moved on and tried to characterize the cargo of these microvesicles via myomic and proteomic analysis. So let's move on to the results. In nanoparticle tracking analysis, we discovered a median size of about 150 nanometers in both samples, and we also detected exosomal surface proteins CD9 and TSG101 in both samples. The figure 1 shows our in vitro assay. So in the bottom left panel, we can see the in vitro mineralization in the control hASCs that are untreated on the left side, and we can see increased mineralization in the SB-treated hASCs. The bottom panel is our treatment group. So on the left side, we see the osteogenically induced hASCs that are treated with microvesicles that are harvested from untreated hASCs. And then on the right side, we can see increased mineralization in osteogenically induced hASCs that are treated with microvesicles that are derived from SB-treated hASCs. The right panel, so figure 1b, shows the alizarin red quantification and clearly shows a significant increase of mineralization after treatment with microvesicles that are pre-treated or that are derived from preconditioned SB-treated hASCs. The bottom panel shows a summary of our gene ontology analysis of our protein molecules. So we can see that there's a clear upregulation of biological processes that are involved in bone regeneration and ossification. Bone-specific protein encoding genes that we identified in these processes are BMP2 and protein kinase A and DDR2. These are important regulators in osteoblast differentiation. DDR2 also encodes for a receptor tyrosine kinase that also regulates bone mineralization and biomineral tissue development. We found many other signaling factors that are involved in the hedgehog and the BMP signaling pathway. So the micromic analysis is still ongoing, and we will report these results later on. So we conclude that hASCs can be conditioned to selectively package osteogenic factors in microvesicles. And this means that microvesicles can be used as vehicles for targeted drug delivery. It can also be preconditioned genetically to improve gene and cell therapy at the target site. And these are non-teratogenic and less immunogenic. they also migrate extensively. So there's a hope that they could potentially replace stem cell-based therapy and circumvent all the risks that accompany this kind of therapy, such as neoplastic transformation. So in the future, we hope to identify more potential osteoinductive microRNA and proteins in the cargo of our microvesicles. We are planning to test them by gain and loss of function experiments in vitro and in vivo. We also hope to add to already existing microvesicle databases and maybe to contribute to off-the-shelf microvesicle ordering. Thank you very much for your attention. I'm also very happy to answer any questions by email. Thank you very much.